So for today we're going to do two procedures. We're going to do aseptic transfers. We're actually going to do two of them. We're going to transfer from your stock unknown that I gave you to a broth and a slant that you just labeled. Then you're also going to do a streak plate, which is a method for isolating organisms. So in addition, on your lab bench, in your group rack is a culture that has the label mix. It's actually a mix of two organisms that we did in a broth culture. That you're going to use the streak plate method to isolate out those two organisms, hopefully. So what do we mean when we say aseptically? What are we trying to do? Not necessarily sterile, because is this room sterile? No, right? So I hate the word sterile technique, although sometimes people say that. It isn't truly sterile, right? But we want to try and keep things from getting contaminated, right? That's your kind of key word here. We don't want your sample to contaminate any of your stuff, which is why once we actually start working, we'll put our notes in our sleeves so that if we do have a spill, which of course happened yesterday, and will probably even happen today, because we're beginners, right? And that's going to happen. Let me know, right, if you spill that broth culture that says mixture. That's a lot of microorganisms. We need to clean it up properly, right? Please try not to. But if you do, right, let me know, and we'll clean it up. Okay, so we don't want to contaminate your culture either, right? We want to keep what your culture is pure, which was the bonus term on the quiz today, right? Pure culture. And so what does that mean? Huh? Oh, it means you got a 10 out of 10 on your quiz today? You're happy. A single species of organism, right? So right now, you don't know what I gave you, but I know, right? Based on your number, what organism we gave you. So we don't want to contaminate it, because then you're going to get false results, right? Because you only want to test one organism. We don't want to contaminate our environment, right? Any of our stuff, especially anything that would leave this room, which is why we limit what comes in and what goes out. So that we want to maintain. So the other thing is that slant in that broth should be sterile media right now. So if you look at them, you should not see any growth, as opposed to that mixed culture. It's nice and cloudy, right? If that was apple juice, would you drink it? Probably not, right? Not a good idea. Your broth tube, right, that you just labeled, it should be clear, right? Does anybody have a cloudy tube? Because if you do, it's probably already contaminated and we need to get you a fresh tube. I usually try to check, but it's always good to have someone else then double check. Remember, always grab your tubes by the tube and never by the cap. Okay, now check your slant. Make sure you don't see any growth on that new slant that you just labeled. Right? As opposed to your green labeled slant, right, which is your unknown, should definitely have growth. You've already should have described that growth. So aseptic just means a condition without contamination, right? So we want to maintain this aseptic. Mixed means just that, right? Is there one organism? No, there could be two or three or, right, several organisms. So slants can get mixed, plates can get mixed, broths can definitely get mixed, right? How would you know that that was a mixed culture? Yeah, the one that's labeled mixed. Right, if you did what we're going to do today and you got different growth patterns, or if you do like we're going to do next week and you get different shapes, then you probably have different organisms, unless it's one that doesn't hold a particular shape, right? So really, other than testing it, just looking at it, if I didn't tell you, you wouldn't know, right? So one of the most important things you do in lab is to label things, right? And label them properly and check your labels as you're working. So we are working with, as far as our unknowns, are pure cultures, hopefully. I didn't screw anything up, or me and Andrew didn't screw anything up. So they're a single species of an organism, right, that you're going to test to figure out what you have. If somehow your culture got mixed, or in the case of the mixed culture for today that I purposely mixed, you could do an isolation technique where you isolate out the individual organisms from a mixed culture. One of the most popular ways in order to do that is called the streak plate method. So you use a plate because it's a large surface area to be able to separate out the organisms. And the method that we're going to use today, we're going to decrease the number of organisms each time we spread them from one section of the plate to the last section. So, and we're going to do this in 4 sections, so the another name for it is referred to as the quadrant streak method that we're going to utilize today. When you see organisms growing on a solid surface, whether it be your slant and your tubes, because they're just easier to store than plates, or your plates, that visible growth you can see is referred to as a colony. Now, is that depending on how that colony arose? If it rose from a single cell, then it's what type of culture? It's a pure culture. If it's multiple cells of the same organism, right, the same species, it's still pure. Can you tell by just looking at a plate or a slant that you have a pure culture? No, you'd have to do tests, right? So again, as we're doing tests, if you see abnormalities, then, right, we may need to assess the situation. We may have some contamination, either from the beginning, hopefully not, right, or along the process. So when those colonies form, especially when we're counting them, we use the term CFU, which stands for colony forming units. So you don't know for sure when you look at a colony if a single cell gave rise to it or multiple cells. So one of the organisms that's actually in that broth tube is Staphylococcus epidermidis. You can figure out how it got its name, right? It's typically found on our skin. It forms clusters of cocci-shaped, spherical-shaped cells. So a lot of times when we're culturing that, we're not actually getting individual cells separated from each other. But we can get a group of them that are all the same species together. And so having known that too, when you're counting bacteria, right, your counts would be a little bit low if you're counting a single colony as coming from a single cell, because that probably wasn't the case for these types of organisms. Streptococci, as you guys will learn as you're doing a homework, form chains, right? And that infection known as strep throat, right, is just a shortening of the name of the organism that causes that infection, right? Streptococcus prurigens causes strep throat. So you're going to start with a pure culture with a single species for your unknown today. It's in a slant, right? So these ones where it's in a tube and you have a slanted surface of agar, that's referred to as a slant, and of course the broths are the liquid. So we're actually going to go from slant into some sterile media, and we're going to go into a broth today as well as a slant. But you could go from any one of these, right, to any other type of media. When we do this, it's referred to as inoculation, right? So when we move the organisms from one place to another, we're inoculating. The actual organism that we're picking up, right, when we pick that up, we call that the inoculum. Now, beginning scientists a lot of times will use a heavy inoculum, which means they pick up a large amount of organisms. Hopefully today I'll teach you not to scrape the entire surface of your agar slant, right? You do not need all of that. If you do that, then you're going to end up with a loop that looks like this one I have as what not to do. And you can really see this one up against the black. It's got crud all over it, right? It's from putting way too much bacteria on this loop. And so you have the ash and the dead crusted on bacteria on this loop. Not what you want to do. Your loop is the instrument that you use, or you could use a needle. The loop, of course, has a loop at the end. And the needles, if people put them back in the correct things, are just that. Oh, this is the straightest needle I've ever seen in one of these. It's amazing. Right, for certain techniques we'll use this. For some of you guys that can't figure out how to pick up a small amount, I'll usually say, why don't you switch to a needle instead of a loop? Because a loop is just not working for you, right? You're just picking up too much bacteria. And so notice this loop too, right? This is a beautiful bend. Yeah, no, not really. You can't use this. So you'll notice even on your checklist today, which is the checklist we use to grade you guys on the midterm, on the back of today's handout, it says the student chose a straight loop or straightened a bent loop. And you want to do this before you start working with your instrument when it's not hot, right? I'm famous for, touching hot things and burning myself. So you want it nice and straight, right, so that you can utilize the instrument properly. This one's a little cruddy. There's something else I wanted to say about this, but it's escaping me right now. So we'll put it away. So, as I said, sometimes people say sterile technique. I'll typically say aseptic technique, right? And then what we want to do is not contaminate things. So we already did our labels, right? We added our number, the temperature, the date, and maybe even our name or initials. We put them up towards the top of the cap, right? I was preparing this at home. I couldn't come into the lab and take pictures of our actual tubes, so it was the best I could find on the internet at the time. I think they work pretty good, but we thankfully don't have these screw cap tubes. Your screw cap tubes were driving me nuts. I was flinging them everywhere. I like our caps. They don't screw on, but that can be a problem too. So you can see a broth and you can see a slant, right? And so the organisms will grow on this slanted surface. These are nice because you can store a whole bunch of tubes, right, very easily in racks as opposed to Petri dishes. And the caps don't come off as easily as a Petri dish lid does. So again, TSA is tryptic soy in the case of A for agar. So it has this component known as a polysaccharide from marine algae. The good news about this component is that we haven't found a microorganism yet that will eat it. And so it's a solidifying agent that we add to the food, in this case tryptic soy. And it allows us to create a solid surface, whether it's the slanted tube or in the case of your Petri dish. These do not liquefy until you boil it, right, the sugar complex. So we can put it at very high temperatures, even the 37-degree incubator, and it's not going to melt. And nobody's going to eat it, right? No organisms eat it as of yet. Not any that we work with or have discovered yet. Unlike gelatin that you might put in your shrimp molds and stuff, anybody know what that's made out of? Protein, which it can be hooves of horses, yes. So it's protein, and just like us, lots of microorganisms like to eat it. So then it doesn't work as a solidifying agent, right, because they're eating it. Also, what do we have to do to get it to solidify? We have to keep it cold, refrigerated, right? At room temperature even, it starts to what? Melt, right? So it's not a good solidifying agent. So we use agar. This was a wonderful invention way back when. So nutrient agar is what we used to primarily use to propagate microorganisms, to grow them. But we found out that that actually isn't the richest or happiest media for them. They actually all do really well. And I know this because all of the organisms, for the most part, I grew on TSA, which is trypticase soy agar. And so this is kind of like baby food for bacteria too, and that the proteins are pre-digested. So the soy tone that's in this is partially digested soybeans, right? So soy protein. Tryptone can be a trypsin digest, which is an enzyme that your pancreas makes, right? So those enzymes are squirted into your small intestine and help you digest your foods. So they use these enzymes to digest proteins. Common protein that's found in these types of media is casein. This is found in milk, and it's actually the protein that makes your milk white. Anyone know the other protein that's common in milk? Whey. W-H-E-Y. Whey. So these are rich medias. They're abundant in nutrients. So beef extract, right, lots of stuff. Peptone is, again, a protein digest of animal proteins. And so food, right? And we collectively refer to all of this food as media, right? Not to be confused with the news media, right? Whole nother beast. So very rich media, right, with lots of food is what we typically use for propagation, just for growth of your organisms. So why do we have to keep moving them from tube to tube? Well, basically those tubes are equated to a fallout shelter with a whole bunch of MREs in them, and the toilet just broke. Would you want to stay there? No. Right? So they're running out of food, and they're sitting in their own swill. Right? They want out. Over time, they're going to get really unhappy. So when you move them from that fallout shelter they're in right now that's gotten really nasty, and you move them to a new fallout shelter, they're really happy, they have lots of food, they start growing until things get really nasty again, right? And they pray you come rescue them again, which is what you're going to do every single week, right? You're going to move them from an older culture to a new culture every week. And we refer to this as subculturing, right? So well-defined media, which I think we only have one that we use in this lab, and it's only because we use it for other properties, is where every single little thing is worked out. How much sugar, how much protein, how much fat, all that stuff is, we know the exact amounts and concentrations and what's there. That's basically equivalent to the Nutrisystem diet for bacteria, which if anybody knows about Nutrisystem, it is expensive. Right? Do we need to spend lots of money for really specialized media and give our bacteria a specific diet? No, we're not doing any type of experiments where we need to know exactly what they're eating. Right? Like I said, we only ended up with one specific defined media, not this undefined complex media that we typically work with, and that's only because we're interested in the other properties that media can sometimes tell us about organisms. So selective media, which those blood agar plates that we used in the beginning to swab the mouth, those are selective plates as well as differential. They select for a particular group of organisms. So propagation is the main reason why we move them, right, from one culture to another. And we could do that with different instruments. Typically we're going to use a loop. If we want to find out a little bit more about the organisms we're working with, or to select for ones that we're potentially wanting to look at, we can use media that does more than allow growth, right? And sometimes, depending on the media, again, you're going to be adding the organisms either with a loop or a needle, depending on how the test is performed. So differential media, usually a wide range of organisms will grow on it, but their growth patterns will differ based on the abilities of those organisms. So it'll help us differentiate one group from another. Just like some of the staining procedures you guys are going to start doing over the next couple of weeks, like gram staining. So a lot of times the differential media that's used in a lab is to differentiate organisms that can ferment certain sugars from others. And because of that, they produce products that can be detected in that media, right? So we see pretty color changes. Or in the case of the blood agar, they make changes to the red blood cells, right? If they have the ability to lyse those. Mm-hmm.
And you put a mixed culture onto the differential. Mm-hmm. A lot of things will grow, mm-hmm. but the fermenting bacteria will do their fermentation, and that lets you know that you have that bacteria in the culture.
Mm-hmm. Yeah, because it's going to look different from the rest on the plate.
Okay. That fermentation process will look different.
Or the media will look different, or the organisms, or both. And you guys will see that later on in the semester. I was kind of hoping. I tried some selective differential, but we didn't get any growth on it, so you guys didn't have anything pretty to look at, except for the mouth swab. But again, the predominant organism predominated that plate. So sometimes too, when you have a mixed culture and you want to separate out particular organisms, you'll use a selective media, and as the name implies, it selects for the growth of particular organisms. And how it typically does this is it inhibits the ones you don't want to grow. So in the case of those blood agar plates, they contain 2 antibiotics that inhibit gram-negatives and allow the growth of gram-positives. Another plate that we use has a very high salt concentration. So any of the organisms that can't grow in that environment will be killed, and we select for the ones that are salt tolerant, right, for that high salt tolerance, actually. Okay, and actually the media that we use later on that's selective is also differential. So you can do both at the same time. You can select and differentiate. But then, of course, the ones you're selecting for, then you can differentiate them into different groups. If you kill it, can you tell if it can do something? No. right? Which is something I'll try to pound in you guys' heads later. Students are like, oh, mine doesn't ferment the sugar. I'm like, yeah, I know, because it didn't. No, you don't know that, because it didn't grow, right? Only if it grew, then you can ask the next question, right? If it doesn't grow, then you can't ask the next question of what it can do. Okay, so why is it important to be aseptic? We want to make sure that we're testing our organism and not some contaminant, right? So the reliability of the tests and keep our environment out. So we want to keep that as pure as possible, a single species. Make sure our sterile media doesn't become contaminated, right? So again, we always check our sterile media before we start, make sure that it didn't become contaminated. Because then when we add our organism, right, we'll mess everything up. Safety. We want to keep these organisms out of the environment. Even though these are non-pathogens, right, they could, for some people, under some circumstances, create problems, right? Any large number of microorganisms could be a problem, right, which we're going to propagate them to large numbers. So we don't want our possessions. That's why we stow them away, right, why we wear protective gear. And to make sure anyone else who uses the laboratory isn't exposed to anything, right? We clean up after ourselves. This is the cleanest lab on campus. Literally the cleanest lab on campus. Because at times it's the dirtiest lab on campus. So for isolation, as I said, we're going to use the streak plate method. So next week after we incubate, you may see plates that look something like these. And so you'll see that they started on one section of the plate with a large number of organisms, and then we got less and less. So much so that hopefully they were separated away from each other, so much that each one of these dots that we call an isolated colony may actually be what type of culture? Pure. It might be pure. And for this one, it all looks the same, right? So maybe it really was all the same, right? Or they only picked up one organism. This one, do you think they're all the same? Probably not, because it's not common for an organism to have multiple colors, right? And we have 3 different colors on this plate. You have this red color, this whitish color, and this yellow. So chances are, right, there was 3 different organisms. Does this mean because they're all one color, it's really all just one organism? No, right? Microscopically or running tests, there might actually be multiple organisms on this plate, right? So it does not guarantee. So we're going to work with a mixed culture from a broth today, but you could take any source, and the easiest way to actually separate them out is on a plate. So you could use a single streak, which is actually what we did, right, when we did our environmental samples, because there wasn't a lot of microorganisms. We just streaked it across the plate, and we actually got stuff to separate. But when you have a heavy inoculum, like what we're going to be starting with today, right, that's a really cloudy tube with a lot of organisms, doing the quadrant streak where we're doing different sections on the plate is going to help us better achieve isolation by reducing the number of organisms as we go. So Peter is kind of fancy. I don't do these diagonal lines. Actually, I don't truly do lines anymore because I've done this enough. I don't need guidelines, right? But we'll have you guys draw some guidelines. You can choose how you want to set up your guidelines for your different quadrants. And I'll give you guys different tips and tricks for that, for setting up your plates. Where are you going to actually draw these guidelines on your plates? On the bottom of the plate where the food is. Never write anything on your lid, right? These things could be separated from one another. So, especially if you're dyslexic like me, it's kind of hard to write the numbers when you're writing them on the bottom, right, so that you can see them when you're looking through the top. So I'll teach you a trick on how to do that so that you can distinguish one number from another and keep track of what way you're going. So what you're going to do in that first section, that first quadrant, is you're going to take your loop and you're basically going to invisible color, right, because you're not going to see the organisms in large numbers until they grow. So you're going to start at the top of your plate with your loop, and you want to try go back and forth, cover the entire first quadrant. Notice it crosses into the second. All the way down, right, to that line border. And you want to cover as much surface as possible, right, invisible coloring. We'll see how well you did invisible coloring after they grow next week. Some interesting plates from yesterday.
So you don't lift it up at all?
You don't lift it up at all. Oh, that's what I wanted to do when I was holding the loop. I just remembered. Okay. So one of the things that you guys need to learn, I probably need a longer one, is what's called a light touch, right? So you want to hold it between your thumb and your pointer finger, and you can even rest it on the next finger, like so you could like draw with it if you needed to, or paint. Most students, when they're first starting out, they push too hard and they bend their loop like this, right? And if it's hot, right, that's when I'm burning myself or you're burning yourself. Because we're going to put these directly into the flame to sterilize them, right? You want to learn a light touch. So even practice, on your hand, your desktop, or something like that, learn that light touch, right? So you're not bending your loop. Okay. Make sense? So then you're going to flame it. So what is that going to do to the organisms that are on that loop? It's going to kill them. So do you go back into that original culture? No, because we want to dilute, right? You only go into that once to put organisms on the first quadrant. That's it. You then flame your loop, and then you're going to be using that loop to pick up organisms from the plate, right? The ones you've already put on the plate that you can't even see, right? Which is why we have the guidelines. So what I do too is to make sure that my loop is cool, because if it's hot and you touch the organism, what do you do to them? You kill them, and you'll know this because it'll go singe, right? And it'll leave a little mark on your agar. So I always touch away from my organisms here at the top of the plate. And then I cross over at least a couple of times. You can do the whole thing. Back and forth, so we're spreading into quadrant 2 and the crossover for 3. When we're done coloring all the way down the line, what do we do with our loop? Flame it, right? Removes all organisms. We go back into the culture. No, where are we getting our organisms? From the plate, from right here on our plate. So now, again, hopefully we're picking up less and less. Now for the next one, and you may even achieve isolation at this step, but we go all the way to the 4th. There's 2 methods you can do. You can pick up a couple times going in, and then just zigzag down the rest of that quadrant. Or what I do, I forgot about this. So sometimes, like when you have strep throat, or you think you have strep throat, they take a swab and swab the back of your throat, right? And then somebody in the lab on a blood agar plate rubs the first quadrant with the swab and then uses the loop for the rest, right? And does a straight plate. But we're going to start out with a loop, not a swab. So I draw mine more like a goalpost, right? Because I can handle that. Straight lines, not diagonal, right? Football Sunday. I'm excited. Dirty birds are going down. Okay. I don't usually even write 4. And I mean, process of elimination, 1, 2, 3. Obviously, the last one's 4, right? What I do, I plugged you in. Oh, maybe I thought I did. There we go. Make it happy. So some people leave out that extra little line right here, right? So they have distinct 1, 2, 3. Do what works for you, right? So 1, 2, 3, and then I do, picking up the loop each time, I do a series of crosses, straight line crosses across from 3 to 4. You can do either a technique, you can try either a technique, but what should you do after you get done or even before you're done? Write it down, right? Which technique did you use so when you come back and look at your plate next week, you can evaluate.
So why is it okay to use just lift it up and do one streak at a time?
Mm-hmm.
That one and not.
You don't have enough space to do that in the others.
There's no
Right or wrong. Yeah. The goal is decreasing the number of bacteria as you move from one section of the plate to the other. But actually picking some up too, from one section to another. So sometimes people just get organisms here and none here, or the rest of the plate, because guess what? They didn't actually pick any up from this first one. That's why you really want to cover the whole surface, so when you're going back in, you're picking up organisms. Does that make sense? All right, good question. Okay. And with practice, you don't need the guidelines, right? You can keep track and go. So sometimes in labs, right, you have, I don't know, a gazillion. Peter will tell you, he's worked with this. You may have 100 samples you have to streak out, You imagine dealing with 100 plates? So they'll do it micro scale. Literally, we call this micro scale. They'll take one plate and divide it into 8 sections. And using sterile toothpicks, they will draw one line with one end of the toothpick, flip the toothpick over, draw another line, right? So crossing one line to another. Take another sterile toothpick, cross another line, flip the toothpick over, and then zigzag the rest of that little section of the plate. So you could do 8 isolations on one plate, doing it micro scale. So you've already kind of looked at growth patterns on for your unknown, hopefully. And there is at least 1 or 2 organisms. This almost looks like there's no growth, right? It's there. It's very translucent, right? So look closely. I've looked closely at most of these, but if you think you don't have growth, call me over and we'll confirm, right, that you have growth on your unknown slant to start with. So some of them are kind of tough. But I mean, you can see this one is nice and chalky, dry looking, where these are more smooth, kind of whitish in appearance. A couple of you guys have some pigmented ones. Actually, I think, who won the pink? Who got the pink? Kelsey got pink? You got pink. All right, that's a new one. I'm excited about that one too. So just one. I just gave out 1 pink one today. So I had a couple of those that grew up from new stock. And then you'll see different growth patterns in broth, so definitely take a look at this, and I'll have you guys do that next week, of your broth tubes that you're inoculating today. You can see the one we have today, right, is really cloudy. It has growth all throughout. It even has a pinkish hue to it if you look at it closely. But some of them will stay at the top. Others might even pellet at the bottom. You want to make notes of these things. Now, on an agar plate, especially, you'll even see sometimes of these different types of growth patterns. We're not going to get this detailed in our recording. I just want to give you guys an idea of what these things could potentially look like on a plate. Is that blurry, or is it just me? I'll need to say something. I think it's just a bad picture. Okay. So, this is good old staph. It's white, right? Not very exciting. Kind of raised, circular, smooth. On blood agar, why are we being specific as to what media it's growing on? Does that matter? Absolutely, right? Growth patterns will even change. Colors will change depending on the media. So this one is one we don't work with anymore, Klebsiella pneumoniae, because it causes a really bad form of pneumonia. But you literally would probably have to lick the Petri dish to get that infection. It doesn't fly off the dish. But these will be relatively white, but you see how they're kind of bumpy, edged, right? They're really shiny. They tend to be very mucoidy. That's on nutrient agar. This is Micrococcus luteus. It's yellow, right? You can guess where it got its name. Luteus means yellow. The colonies, if you were to look at them, are convex, right? They're like little hills. They're circular in shape. They're nice and smooth. This is on brain heart infusion. These are, even as time goes on, right? 24 hours versus 48 hours on blood agar. Bacillus subtilis. Notice how rough it is and how you get these flat edges, right? It's very dry looking. Some of you guys have ones that look like that. So you want to make sure you make a note of that.